In this last lecture, we're going to talk about a second technique that we can use to measure the rate of photosynthesis. The first one was using the floating disk method. The second technique, or the second method, is sometimes called the reduction technique. Sometimes it's called the dye reduction technique. So let's indicate that so you're not confused if you hear it referenced as the dye reduction technique. Now, instead of doing this actual lab in class, we're going to run it virtually. And it's basically because we're kind of out of time and we can do it much faster by doing it on a computer. Now, before I get into this technique, I just want to point out that this lab in which we use the dye reduction technique, we used to have to do it with the old AP curriculum. It was one of the 12 labs that were mandatory in the program. Now remember, they changed the program about 3 years ago, and we have options now for labs that we can do. So the AP lab manual now actually has us utilizing the floating disk method. But I've seen too many AP questions in which they're talking about this dye reduction technique. And if you haven't been exposed to it, it's really hard to answer those questions. So again, we're going to go through this dye reduction technique. Now, I will say that I feel like you guys are going to understand this dye reduction technique better than in the past years, because I think you have a very good understanding of what we use a colorimeter for and what we mean by absorbance and transmittance. And those terms are related to this dye reduction technique. Now, you can probably guess from the name of the technique is that we're going to use a dye to help us measure the rate of photosynthesis. And this molecule that we're going to use is called Dpip. So here's a little bit of background about Dpip. Dpip is a very beautiful blue color. If Dpip gets reduced, that means it can accept electrons, it's going to turn clear. So we can measure the change in color over time, and then that's going to give us a rate for photosynthesis. Let's take a look at this diagram and how Dpip relates to some of the processes that we see happen in photosynthesis. Now, what I'm showing you here is a piece and a cross-section of a thylakoid. Remember, they look like coins, so it wraps all the way around. They're just showing you a little bit of it. Remember, embedded in that thylakoid membrane is the electron transport chain. So let's go ahead and label it. That's the electron transport chain. So here I've added Dpip, and Dpip, like I said, is blue before it gets reduced. Now remember, in the light reaction, light is being absorbed, it's exciting electrons, and electrons are being passed down the electron transport chain. So let's show the electrons being passed down, and eventually this electron arrives at this last protein in the electron transport chain, and it can't go anywhere. And that is where the molecule NAD P+ gets involved. It's a final electron acceptor. And when it accepts that electron, it's reduced and it forms that NADPH molecule, which is then going to go to the Calvin cycle. But I'm not going to show the NADP+ coming in and accepting that electron. That's now the job of Dpip, this blue dye that we're going to mix in with our chloroplast suspension. So Dpip comes in, and it's the final electron acceptor. So show that it is gaining an electron, and whenever it is reduced, it becomes clear. And that's what this diagram is trying to emphasize. So if we have something where there's greater light intensity, we're going to have a greater amount of electrons being excited. So we have more Dpip that's getting reduced, and we're going to see a faster change in color from blue in our cuvettes that we're going to set up here to clear, indicating there was a high rate of photosynthesis if we have a high rate of color change. So Dpip is basically taking the place of NADP+. And again, the higher the rate of photosynthesis, the greater the amount of excited electrons. The faster Dpip is going to be reduced, changing it from blue to clear. So let's talk about this lab setup. We're going to use cuvettes, and we've used cuvettes before because we've used cuvettes in the colorimeter. And in the cuvette, we're going to put some chloroplasts. Now, I can isolate chloroplasts by putting spinach leaves in a blender and pulverizing those leaves and basically breaking up the cell membrane to release the chloroplast, I can then take that solution and I can put it in a centrifuge, and it's going to spin it. And what it's going to do is it's going to spin the heavy things at the very bottom of a test tube. And so I'm going to have chloroplasts at the bottom of the test tube, and I can suction them up, and I can take those chloroplasts and I can put them in this cuvette. So that is what I mean by a chloroplast suspension. Basically, we're going to pulverize some spinach leaves and take out the chloroplast. We have to put a pH buffer because pH isn't going to be regulated anymore in the environment of these chloroplasts because we've destroyed the cell. And really the cell is just trying to help maintain this pH balance. So we have to put a buffer in here. So the buffer is going to go into each of the cuvettes. And then we have to add the Dpip. Now, this is where you might want to pause the lecture and grab a dark blue colored pencil so that we can shade in these cuvettes so you can see basically what we're looking for as we do this lab on photosynthesis. So as soon as I put the Dpip in the cuvette, then it's going to turn it very, very dark. And we also have water in here, distilled water. So let's go ahead and let's shade this really, really dark. And it's really, really dark because of the Dpip dye. If I now take this cuvette and I put it in a colorimeter, I can measure the transparency. And if you think about it, not a lot of light is going to make it through this dark dye. So it's going to have a very low transparency. So starting off, we're going to have this low transparency. And let's say I'm taking my cuvette now with my chloroplast and I put it in front of a light so that photosynthesis can occur. So as long as light is being absorbed, then we're exciting electrons and we're going to reduce this Dpip. So think about after 15 minutes, what should be the new transparency if photosynthesis is in fact occurring in these chloroplasts in the cuvette. Now remember, as Dpip is being reduced by photosynthesis, it's becoming more clear. So take your blue colored pencil and you can just barely shade it in. Just make it a light blue, and let's think about transparency. Light's going to go through that. So we just made that solution more transparent. So our transparency can go up to something, let's just say 68%. Now hopefully just from this information that I've given you, you know how you could calculate the rate of photosynthesis that was taking place in this chloroplast suspension. You have the change in something. So we have the change in percent. We have an indication of time that happened in 15 minutes. So our change in transparency is 30%, and that happened in 15 minutes. So we're changing 2% per minute. And that's a rate for photosynthesis. Okay, so let's set up another scenario in a different cuvette. So I'm going to isolate chloroplasts in the same method that I described before, and then I'm going to boil them. And after I boil them, I'm going to place them into another cuvette. Again, I have to put that pH buffer in there, and I have to add my dye that can be reduced, that Dpip. And as soon as I put the Dpip, then again, it's going to be a really dark, beautiful blue color. With this information, think about how the transparency of our cuvette is going to change if we sit it in front of a light for 15 minutes. Hopefully you understand that if you boil the chloroplast, then you're going to denature all of the proteins, like the proteins in the electron transport chain, all the enzymes that are helping to fix CO2 and reduce it in the Calvin cycle to form sugars. All those enzymes are going to be denatured, so photosynthesis is not going to occur. Think about what would happen to these proteins if you are boiling the chloroplasts and the thylakoids that are inside the chloroplast. So they're going to change shape, and then they're not going to be able to function. So if we put the boiled chloroplast suspension in front of light, we're not going to have any exciting of electrons and pass them down the electron transport chain. So we should not see any change in transparency. It should be still at about 38%, and it should be just as dark as what it initially started off of. So again, we can calculate the rate. This one will be an easy one. Our change in percent is 0%. Our time was 15 minutes. So we can see that the rate of photosynthesis equals 0, or 0% per minute. Now, hopefully you can look at data like this now and you can explain the results. So you see that we have basically 3 cuvettes that were set up. So this cuvette was exposed to light, it had unboiled chloroplasts, and every 5 minutes we are measuring how dark this sample is. So remember, when we first put in Dpip, at time 0, it should be very, very dark. It's going to be that really dark blue color. And if photosynthesis is occurring, then that Dpip is getting reduced and it's turning more clear over time. So more clear over time. So we can tell that photosynthesis is occurring, and we can calculate rate. So how would you go about calculating a rate of photosynthesis for this first cuvette? Again, it's just change in something over the change in time. So my change is from 28.8 down to 66.7. So that's going to be my top number. My change in time in this situation looks like it's 20 minutes. So you should be able to explain these results and tell me why it started out blue and it ended up more clear. You should also be able to look at these data points and be able to calculate rate of photosynthesis. Let's take a look at sample 2 and try to explain those results. We can see that we really haven't had much of a change in transmittance over time. So it looks like we have a very low rate of photosynthesis. So in this situation, we can tell that's because we're not getting any electrons excited by light, because this cuvette was placed in the dark. So if there is no excitement of electrons, there's no electrons to reduce the Dpip. So that cuvette is going to start off dark blue with very low transmittance, and it's going to stay dark blue because that dye is not getting reduced due to the light reaction. And then our last cuvette, we have light, but we have those boiled chloroplasts. So again, we're starting off a very, very dark blue, and it's going to end pretty much in the exact same spot because we've destroyed the proteins that are necessary for photosynthesis to occur, for those electrons to become excited and get passed down the electron transport chain to reduce the Dpip. Now take a second to see if you could draw some lines on this graph, and this graph is showing a change in percent transmittance over time. And let's see if you can add 3 lines that would show the rate of photosynthesis if you put some chloroplasts in a cuvette and then you heated it up to 100 degrees Celsius. You put some different chloroplasts in a cuvette and you had it at 30 degrees Celsius. And then if you cooled off the cuvette with chloroplasts to 10 degrees Celsius. So we have 3 different cuvettes in 3 different situations. See if you can draw the 3 lines on this graph. Before I draw the lines, I just want to note that we have percent transmittance. We have a change in something and then over time. So technically, rate is shown by the slope of the line. So let's talk about the cuvette that was put in the 100 degrees Celsius environment. We know that photosynthesis isn't going to occur. So our transmittance is going to start out, it's going to start out blue. So let's start it up here. Let's think of this as that being about that 38% that we used in the example. And if photosynthesis isn't occurring, then we're not going to have a change in transmittance. So slope equals 0. So rate equals 0 as well. For the 30 degrees Celsius, that is slightly warmer than room temperature. So we've heated up this cuvette. And if we increase temperature, then that's going to increase reaction rate. So to show that, we need a line that's going to show a steep slope. So again, we can start it out at 38% transmittance, and it's going to become more clear over time, or more transmittant. So we can show rate like that. We have a high slope, so that means we have a high rate of photosynthesis. And let's talk about the one that is chilled down to 10 degrees Celsius. Again, we can start it out at 38% transmittance, which is going to be a dark blue when we add that Dpip. Now we're still going to have photosynthesis, but remember, reaction rates and all the different steps of photosynthesis are technically reactions that are taking place. The reaction rates are going to be lower. So we're going to start out with a blue cuvette, and it is going to change to clear, but it's just going to take longer to do that. So maybe by the same amount of time, it's only changed the transmittance from here. I feel like a lot of students would put some kind of negative sloped line coming down, and that's not the case. We're still going to change the transmittance, make it more clear. So let's put this at about, I call it 50 if you want. So let's say it's at about 50% transmittance. And eventually it will go all the way clear. It's just going to take a lot longer time for all of the Dpip to be reduced. And with that information, you should be able to set up and run this dye reduction technique virtually and be able to draw conclusions and understand what's going on when we use this technique.